Hello everybody. In this video, we are going to discuss the UV visible spectroscopy for competitive examinations like a CSIR, GATE, IIT JAM, TIFR and TRB examinations. So I have divided this discussion into 5 parts. So in 4 parts, I will be discussing the important terminologies involved in UV visible spectra and Woodward-Fisher rule for calculation of lambda max. And in the final part, that is part 5, I am going to discuss the questions asked in CSIR, GATE, IIT JAM, TIFR and TRB examinations on UV visible spectroscopy. So the first part, let me discuss. So the spectroscopy, it is nothing but the study of interaction between the matter and electromagnetic radiation. So we know what is electromagnetic radiation. So I will explain. So it is known as EMR, and it is any form of radiant energy that is propagated as a transverse wave, and it vibrates perpendicular to the direction of propagation, and as the name suggests, it possesses both electrical and magnetic field. Look at this, you can see. So the distance between two maxima, you can see, so this is a maxima. So the successive maxima, so the distance between two successive maxima is called wavelength or the minima, you can see. So this is a wavelength, it is denoted as lambda, and you can see, so this is electrical field oscillation, this is magnetic field. So both are perpendicular to each other to the direction of propagation. And it possesses a certain amount of energy, as I said earlier, and the energy ultimately is an indivisible unit of radiation, that's called a photon, and that photon is related to the frequency or wavelength by the equation. So this is a famous equation, E is equal to h nu, and nu is a frequency, which is further denoted as c by lambda, that is c is velocity of light, lambda is a wavelength. You can see energy is inversely proportional to wavelength. Okay. H is the Planck constant, nu is the frequency, c is the velocity of light, lambda is a wavelength, and this is electromagnetic spectrum. You can see in the left hand side this is gamma rays, and in the right hand side you can see radio waves. So as from left to right, you can see the wavelength increases. It's a lambda values increases from left to right, that is gamma rays to radio waves, and the energy decreases because the wavelength is inversely proportional to energy, I can see. So hence radio waves possess a longer wavelength and gamma rays possess a shorter wavelength. So this is very important because there are so many questions asked in competitive examinations, so which is having a shorter wavelength or which is having a longer wavelength. You can see radio waves possess longer wavelength, and you can see gamma rays is shorter wavelength. And when photon hits a molecule, what happens? So it is absorbed, the photon is absorbed by the molecule, and the molecule is promoted into a more excited energetic state. And you can see, so ultraviolet to visible light has enough energy to promote the electrons to higher electronic state, that is from the HOMO to LUMO. HOMO means highest occupied molecular orbital to lowest unoccupied molecular orbital, and these differences between HOMO and LUMO is called band gap. You can see, and this is HOMO, highest occupied molecular orbital. You can see occupation of pi electrons. So this is an example of ethylene, and you can see LUMO. And the energy gap between HOMO and LUMO is called band gap. And when the molecule absorbs a photon, so one of the electrons goes to the higher excited state, that is pi to pi star. This is called pi to pi star transition. And you can see in the case of butadiene, you can see there are 4 pi electrons. Okay, these two are Psi1, Psi2. This is actually HOMO, highest occupied molecular orbital, and this is LUMO. LUMO means lowest unoccupied molecular orbital. If there is no electrons, and if excitation takes place, the one of the electrons in Psi2 goes to Psi3. So the energy gap between these two, Psi2 and Psi3, is 215 nanometer compared to, you can see, this is 185 nanometer. So the energy gap is maximum in ethylene, and as the conjugation increases, you can see the energy difference decreases. So we'll discuss later. So the alternative name for UV visible spectroscopy is electronic spectroscopy, because it involves the promotion of electrons, sigma electrons, pi electrons, or non-bonded electrons from the ground state to the higher energy state. You can see the regions of ultraviolet visible spectroscopy. So between 10 and 200 nanometers known as far or vacuum UV, and 200 to 380 known as near or quartz UV, and the visible region is 380 nanometer to 780 nanometer. And the ultraviolet visible region has further divided into three. It's ultraviolet A, ultraviolet B, and ultraviolet C, depends on the lambda max value. You can see, so ultraviolet C is a very dangerous rays. It has been filtered in the atmosphere itself, and you can see the range of UVA and UVB. So the instrumentation, so the main source is ultraviolet range. You can use a hydrogen or a deuterium lamp. For visible range, tungsten lamp has been used as a source. And the filters, there are two types of filters are available. That's absorptive filter and dichroic filters. That is a reflective filters, and the monochromator. So the role of monochromator, it produces a beam of light with a very narrow bandwidth or light of single color. It allows only a particular wavelength. So mono means single, chroma color, and the sample cuvette. So cuvette is normally made up of a quartz window, because the glass is not transparent to ultraviolet radiation. So it will absorb some amount of radiation. Hence, glass cannot be used as a sample cell or cuvette in ultraviolet spectroscopy. So quartz has been used. And the detectors, the commonly detectors are photo tube detectors, you can see, are photo multiplier tubes, or silicon diode array, or charged coupled device array. This is common detectors. And you can see, so this is a deuterium lamp, this is tungsten lamp, act as a source. And so the role of filters, can filters to the monochromator. Here the monochromator allows a single wavelength. You can see the single wavelength, and it reflected to the beam splitter. And here you can see, so this is a sample cuvette, and this is a reference cuvette. So after passing through the monochromator, the light passes through both reference and sample cell, and here you can see the photodiode, that is a detectors, detect and processed, and will get the UV visible spectra. This is instrumentation. Here electronic excitations, you can see, so this is sigma electrons, pi electrons. This is non-bonded electrons. They are not involving any bonding. And you can see these two are anti-bonding, pi star and sigma star. These are all different types of transitions. And here, so from the figure you can conclude that, so the sigma to sigma star, you can see sigma to sigma star electron requires higher energy, because you can see the energy difference between sigma and sigma star. And this is the increasing energy order of electronic excitations. So you can see sigma to sigma star electronic transition requires higher energy, and n to pi star transition requires lesser energy. This is very important, because so many questions have been asked regarding the electronic excitation energy level. You can see, and this is examples for sigma to sigma star transition takes place in alkanes, and n to sigma star transition, that is non-bonded atoms should be there. Hence oxygen, nitrogen, or sulfur possess non-bonded electrons, and halogens and halides can appear n to sigma star transitions, and pi to pi star transition occurs in So sorry, unsaturated bonds, that is alkenes, alkynes, aromatic rings are carbonyl and azo groups, and N2 pi star transition occurs in the carbonyl group because it has both non-bonded electrons and pi electrons. And this is a pi 2 pi star transition. This is N2 pi star transition. So normally N2 pi star transition occurs between 270 and 300 nanometer. For carbonyl group, it shows presence of aldehyde or ketones, and you can see. So this is absorption maxima for common solvents used in UV visible spectroscopy. You can see acetonitrile appear to 190 nanometer, for water 191 nanometer. So this is a commonly used solvents in UV visible spectroscopy. Thank you so much. So please subscribe to my YouTube channel for more videos. Thank you again.